Hello everyone, dear students. We have completed meristematic tissue in our previous lecture. Now in this lecture we are starting with the permanent tissue. We are starting with the permanent tissues. Okay, so permanent tissue are what? First we should understand what are permanent tissue. So these are derived from meristematic tissue, derived from meristematic tissue, and these are the cells who lost their capacity of division. Those cells who have lost their capacity of division, they are known as permanent tissue. Means permanent tissue have a cells. So permanent tissue consists of non-dividing cells. It consists of non-dividing cells. Now these cells or non-dividing cells can be living or dead cells. So means permanent tissue composed of living or dead cells. They are primary permanent tissue. They are of two types. This permanent tissue are of two type, that is primary permanent tissue and secondary permanent tissue. This will be called as a primary permanent tissue if they are derived from apical or intercalary meristem. If derived from apical or intercalary meristem, whereas secondary permanent tissue are derived from lateral meristem. They are derived from lateral meristem. Means this is a type based on origin, from where or from which type of cell they are originated. Based on that they will be called as primary or secondary permanent tissue. Now this is the first classification. Next classification of permanent tissue we are going to talk about, that is based on the structure and function. So next, permanent tissue, they have a specific, one more important point that you remember, that permanent tissue are known as mature tissue, because meristematic tissue are immature, because meristematic cells are considered as immature cells. Now this permanent tissue or mature tissue are of three types. First are of two types actually, simple tissue and secondary complex permanent tissue. Now in the simple tissue we will study about parenchyma, collenchyma and sclerenchyma. But when we talk about complex tissue we will talk about xylem and phloem. We will talk about xylem and phloem. So now we will talk about first simple tissue, then we will talk about a complex tissue. So first we start with simple permanent tissue. Now the simple permanent tissue are made up of one kind of cell. They are made up of one kind of cells performing similar function. If they are made up of same kind of cell and perform a similar function, then they are called as a simple tissue. Now this simple tissue are of three types, that is a parenchyma, collenchyma and sclerenchyma. We are going to study this parenchyma, collenchyma and sclerenchyma one by one. So first we start with parenchyma. When we talk about parenchyma, this parenchyma are found almost in parts, found almost in all parts of plants like roots, stem, leaves, fruit, seeds. So in such type of almost all parts there is a presence of parenchyma. When we talk about a characteristic of parenchyma are cells are isodiametric. Its cells are isodiametric. They are made up of similar size of cells. Shape of this parenchyma cells can be spherical, oval or polygonal. Then intercellular spaces may or may not be present. Intercellular spaces may or may not be present. When we talk about them, they are living tissue, and if they are living tissue means they have a thin cellulosic cell wall. They have a thin cellulosic cell wall. Sometimes they are elongated, then this elongated parenchyma with tapering ends is called prosenchyma. It called as prosenchyma. So be remember, what are prosenchyma? Prosenchyma are those cells, those parenchymatous cells which have a elongated and tapering ends. Now this is the part with respect to the structure. Now when we talk about the function of parenchyma, the main function is of the storage of food. So main function is storage of food. Okay, example is fruit and endosperm. In this parenchyma are present which stored food. Next is secretion. Next function is secretion, and sometimes they store secretory or ergastric substances. Store secretory or ergastric substances such as tannins, resins and gums. This type of cells which store this, they are known as idioblasts. So idioblasts are nothing but a parenchymatous cells which store secretory or ergastric substances. Okay, so I hope you got this. This parenchyma are of two types, that is Parenchyma and parenchyma. Now, if they are of parenchyma, then when parenchymatous have chloroplast, when parenchyma with chloroplast are known as parenchyma. So what be the function of them? Function become photosynthesis. But parenchyma are parenchyma with large air cavities. Parenchyma with large intercellular spaces known as air cavity. So if they have an air cavity filled with air, and that is a large intercellular spaces, they are known as parenchyma, and they help in function is help in circulation of air and provide buoyancy to plant. Buoyancy is a thing of floating, so it provides a plant to float in the water body, that is due to the presence of this parenchyma. Now we start with second type of simple permanent tissue, that is parenchyma. Okay. Now parenchyma term is coined by Sladen. Term is coined by Sladen. Now when we talk about parenchyma, the cells of parenchyma have thickening on the cell wall, have thickening at corners of cell wall. Okay, so at the corners of cell wall, in this thickening is found. Okay, these are living elastic mechanical tissue. They are mechanical tissue, but they are living and elastic. Elastic means they are providing a flexibility. So which is a flexible tissue? If they ask that, that's a parenchyma. They are not found in root and monocot stem. Important statement that you should mark. They are not found in root and monocot stem. And monocot stems. These cells form hypodermis of cell. It form hypodermis of stem, and they are also found in petiole. Now their cell wall are thickened, but cell wall thickened due to the deposition of the pectin and cellulose. So cell wall contain high amount of pectin and cellulose. So their cell wall is made up of pectin and cellulose. In this, lignin is absent. Why? Because lignin usually found in case of dead cell. In case of living cell, lignin deposition is not found. Now this parenchyma are of three types. This parenchyma are of three types. Number one, first type is angular parenchyma. They are angular parenchyma. Angular parenchyma are those in which angular walls are thickened. In this, angular walls are thickened. So if we draw a diagram of this parenchyma, then you will see that they have some cells like this, and in these cells you can see they have a wall thickening, but their angular walls are much thickened. I am showing this here with red cell, with red marking. Here you can see their angular walls are thickened in the cell wall. Be remember, not in a plasma membrane, in a cell wall. So if these angular walls are thickened, then they will be called as angular parenchyma. And within that you will find that plasma membrane, nucleus, all these things you will find within that. So they have a angular thickening, and this angular thickening is responsible for their nature. Now if you see the example of this angular parenchyma, so angular parenchyma is found in stem of marigold, tomato, and datura. These stems have this angular parenchyma. Second type is lamellet parenchyma. Second type is lamellet parenchyma. Now this lamellet parenchyma are those in which tangential walls are thickened. Tangential walls are thickened. So if we draw this type of diagram of a cells, so if we see a lamellet thickening, the lamellet thickening are found on the tangential walls, means it is found here. Whole tangential wall are thickened. And these tangential walls are much thickened. Because of this, it is known as lamellet parenchyma. Okay, so these are the lamellar thickening. These are lamellar thickening which are found. Because of this, they are known as lamellet parenchyma. Now this lamellet parenchyma are found in stem of sunflower. So the example is of lamellet parenchyma is sunflower, stem of sunflower. Now next, third type is lacunate parenchyma. Third type is lacunate parenchyma. Now lacunate parenchyma are those which have lacunate thickening. Now lacunate means a lacuna, means thickening on sides of intercellular spaces. Wherever you find a intercellular spaces, at that part you find a specific thickening, then that is known as what? A lacunate cholenchyma. Now the example, if we see, example are considered in this, that is a stem of calotropis. Okay, so if we want to draw this, we are drawing some cells. Suppose these are the cells, okay, and these are the intercellular spaces. So thickening become more on the intercellular spaces side, like this. So more on the intercellular spaces side, then it is known as a lacunate cholenchyma. So I hope you understood the three types of cholenchyma, that is angular, lamellate and lacunate. Now we talk about function of cholenchyma, and the function of cholenchyma is to provide mechanical support, flexibility, elasticity to organ, and due to peripheral position in stem, they also resist bending and pulling of stem or petiole during action of wind. So it is useful for the young plants and herbaceous organ, where it is important supporting tissue or mechanical tissue, and it is also pulling or bending of this petiole is importantly due to this elastic tissue, that is cholenchyma. Okay. Now after that, next we start with the sclerenchyma. Third type of simple permanent tissue are sclerenchyma. And when we talk about this sclerenchyma, they have a thickened secondary wall. They have thickened secondary walls due to deposition of lignin. These are a dead cell, non-living cell. And they become dead or non-living at maturity. On maturation, they get dead. The cells have a simple pit. They have a simple pits. Pits are unthickened areas of secondary cell wall. This sclerenchyma are of two types. One is sclerids and another one is sclerenchymatous fiber. Second is sclerenchymatous fiber. First we talk about sclerids in detail. Okay, first we talk about sclerids in detail. When we talk about sclerids may be spherical, oval or cylindrical. They can be spherical, oval or cylindrical. They have lignified and extremely thick wall. They have a thick wall. Okay. And due to lignin deposition, more lignin deposition, and due to a more cell wall formation, the lumen is almost obliterated. Lumen is almost obliterated. Obliterated means it is almost reduced. Okay, this type of lumen they have, and they are found in hard parts of the plant. They are found in hard parts of plant. So if we see the examples of them, they are commonly found in fruit walls of nuts, fruit wall of nuts, pulp of fruit like guava, pear and sapota. Then in seed coat of legume and leaves of tea. This provide mechanical support. They provide mechanical support, and they are of several types. So next talk about types of sclerites. In this, first type is brachysclerites. Brachysclerites are also known as stone cells. They are also named as what? Stone cells. So if we talk about them, they are isodiametric and they are found in bark with phloem. They are isodiametric and found in bark with phloem, cortex, hard endocarp and fleshy portion of some fruits. So they look like this. So if we draw the diagram of this brachyscleride, in this brachyscleride we can see that their cell wall is so elongated and the cell wall have a pits. Okay, so this is the cell wall. So this much thick cell wall is found. You can see this much cell wall is found in case of this brachysclerids. And in brachysclerids, this elongated structure are named as pit canal, where secondary deposition is absent. They are provide grittiness to fruit like guava and pear. They also provide a grittiness to this. So this is the first type, brachysclerid stone cells, and they are hard. Now after that we talk about next type, second, that is macrosclerids. So macrosclerids are also known as rod cells, and this rod cells somewhat look like this. They look like this. Okay, so then they have this particular lumen. So if you talk about macrosclerid, their roles are they are elongated cells, rod-like, and found in seed coat of leguminous plant. Found in seed coat of leguminous plants. Okay, next, third type is osteosclerids. This osteosclerids stands for bone. Osteo stands for bones, so they are named as bone shaped or bone cells. So they look like something this. So these bone cells appear like this osteosclerids. They are rod-like with dilated ends. They have dilated ends and they are barrel shaped. They are found in, if we see example, leaves and seed coat of many monocot and seed coat of subepidermis in legumes. Seed coat of subepidermis of legumes. Their osteosclerids, bones type of sclerids are found. Fourth is asterosclerids. These asterosclerids are star cells because they look like star. And if we draw their diagram, they look like this. So because of this, they are named as star shape, and they have a thick wall like this. The star shape they will be found. So these asterosclerids they are star shaped, and if we see the example, they are common in petiole of floating hydrophytes petiole of floating hydrophytes like nymphia, lotus, and leaves of tea. So in this, these asteroskeleton are found. Then we talk about the fifth one, that is trichoskeleton. When we talk about this trichoskeleton, they are internal hairs. They are internal hairs. So in this, if we see the diagram somewhat look like this. So this is lumen. The lumen is unwitched and they have a hair-like projection here. So they have long hair-like branched skeletons. These are long hair-like branched skeletons, and common in hydrophytes and aerial roots of monstera. So that is the localization of this trichoskeleton, and these are all types of all type of skeletons. Next we talk about the sclerenchymatous fiber. Sclerenchymatous fibers, if we talk about the characteristic of that, they are long with tapering ends, These are thick walled cells. They have a lignified thick wall. Okay, they are fully developed fiber cells are always dead. The length of fiber varies from 2 to 50. Length varies from 2 to 550 mm in angiosperm and 1 to 12 mm in gymnosperm. These sclerenchymatous fibers are present in hypodermis of monocot stem, pericycle of many dicot, and in secondary wood and in vascular bundle sheath in monocot stem. This sclerenchymatous fiber, if we take an example of this monocot stem, then jute, flax and hemp. Okay, if we talk about the living sclerenchymatous fiber found in tamarix, and the function of sclerenchymatous fiber is to provide a mechanical support. So function is to provide a mechanical support. And if we see the diagram of this sclerenchymatous fiber, then it look like somewhat this. and lumen is almost reduced, and they can have a septa as well. Suppose this is the lumen, then they have a septa. So this fiber can be septate or aseptate. So that is what about sclerenchyma fiber and sclerenchyma. So here simple permanent tissue get complete. Now next we will start with the complex permanent tissue. So now we start with complex permanent tissue, and this complex permanent tissue we talk about xylem and phloem. So first we will talk about xylem. First we talk about xylem. This xylem term given by Nageli, okay, but this xylem is also known as hedrome, by Haberland. Okay, by Haberland it is known as hedrome. So if we talk about their characteristic, it is a chief conducting element, chief water conducting element. It is a chief water conducting element, but it also provide mechanical support. Now on the basis of origin, this xylem is of two type. Xylem is of two type on basis of origin, that is primary xylem and secondary xylem. When we talk about primary and secondary xylem, primary xylem is derived from procambium and it is formed during the primary plant body. Okay, so it is primary xylem is formed during primary growth or lengthwise growth. Okay, now this primary xylem is formed initially. It is again differentiated into this primary xylem is differentiated into protoxylem and metaxylem. What it is differentiated? Protoxylem and metaxylem. Now this protoxylem and metaxylem, if we talk about, this protoxylem are the first formed primary xylem, whereas metaxylem are formed later. Okay, then this protoxylem consists of tertiary element, means tracheids and vessels, and Xylem Parenchyma. But when we talk about the specifically of this metaxylem is laterally formed, but it consists of tertiary element Xylem Parenchyma and xylem fiber. So it is the first form and later form. Metaxylem are bigger in size and protoxylem are smaller in size. So please remember the cells of metaxylem are smaller in size, whereas of this metaxylem are bigger in size. The cells are bigger in size. So that is all about what primary xylem. Next we talk about secondary xylem. So secondary xylem is formed from cambium during a secondary growth. So it is formed during secondary growth. It is formed during a secondary growth. Okay. And it is differentiated into two systems. The first one is axial or vertical system. Axial or vertical system. This axial or vertical system consists of tertiary element, xylem fiber, tertiary element. It consists of tertiary element, xylem fiber, and Xylem Parenchyma or wood parenchyma. Whereas second is ray or horizontal system. Ray or horizontal system. This consists of ray parenchyma for storage of food. They consist of ray parenchyma, so which help in radial conduction as well. Okay, so this is the primary xylem. In this protoxylem, metaxylem, and secondary xylem. Secondary xylem during the secondary growth, primary xylem during the primary growth. So xylem, we have studied about the primary xylem and secondary xylem. Now we should talk about the components of the xylem. First we should talk about the component of xylem. So xylem is a tissue which consists of 4 types of cells. In this, first is xylem tracheids, second is xylem vessel, third is xylem parenchyma, and fourth is xylem fiber, and last is xylem parenchyma. Now these 3 are non-living or dead, but this is the living component or living cell. Now we are going to discuss about them all in one by one. Okay, so first we start with the tracheids. So first is tracheids. So tracheids are elongated cells with tapering ends. They have a tapering end, or chisel like ends are there. They have a thick wall, that is lignified cell wall, cells loss protoplasm and become dead at maturity. So these are the tracheids. Now if we talk about tracheids, which is a particular NCERT diagram, so you can see these tracheids look like this, and another tracheid is arranged like this. So they have a thick wall, lignified cell wall, okay, but they have pits. They have some pits. So through these pits, the transfer of water occurs from the lower part to the upper part that occur through the pits. So one cell absorb water and transfer it this. So these tracheids are unicellular. These tracheids are unicellular and they have a narrow lumen. Why they have a narrow lumen? Because that is due to loss of protoplasm, the lumen get reduced. So these are the structural part. Now we talk about function. So first function is the transport of water, that's a conduction of water and mineral. But when we talk about another function, that is a mechanical support, is also provided by these tracheids. Why? Because tracheids have a thickened cell wall and thickened cell wall play a role in mechanical support. Now lignin deposition in tracheids is of various type, and based on lignin deposition we call that tracheid, we give a particular name to that tracheid. So first is annular, second is spiral, third is reticulate or net-like, fourth is scalariform, that is ladder-like. So if we talk about a first, that is a annular, in this when we see the tracheids, in tracheids internally you will observe that they have a lignin deposition like this, annular. When we talk about spiral, in spiral the lignin deposition is like this. And reticulate is net-like. So in this, if we see, you will see a whole net-like structure in case of this. That is net-like reticulate. And the last one, that is a scalariform ladder-like. So in this, lignin deposition is like this. So this type of ladder-like lignin deposition is found in case of this is scalariform. And the fifth type is pitted type, in which we will see the arrangement of pits in tracheids. So that is known as pitted type. So this type of tracheids are found, and tracheids are obviously a primitive conductive element that we know. Now after tracheid we talk about the vessels. So second tracheidy element are the xylem vessels, and xylem vessels are also named as tracheae. Be remembered, tracheae and tracheids are different. Tracheae are vessels, whereas tracheids are the particular unicellular structure. Now this tracheae or vessels, they are multicellular. They are made of several xylem vessel element. They are what about xylem vessel element. Okay. Now they are elongated, and they are tube-like. They are tube-like, and here cells are placed one above the other. Okay, so these are the particular characteristic. Their walls are thickened and lignified. They dead at maturity. They are dead at maturity and without nuclei. Okay, so this is the thing. So if we see its structure, so when we draw the structure of vessel are elongated tube-like structure, and they have a, so here they have some oblique divisions. Okay, we call them septa. So this is, if you see, this is one xylem vessel element, and xylem vessel element are placed one above the other. Now they have a thickened cell wall. Okay, but when we talk about this, the cytoplasm is reduced, so cytoplasm is present on the periphery. Why? Because internal part is using conduction, and they have here perforation plate which help in conduction. So these are perforation plates. So one above the other xylem vessel element are arranged. Okay, so if we talk about the xylem vessel element or xylem vessel are present in angiosperm and absent in pteridophyte, gymnosperm, and some primitive angiospermic families like Winteraceae, Trochodendraceae, and Tetracentraceae. These three types of families are there which lack these particular vessels in angiosperm also. Means in all angiosperm these vessels are present, that is not like that. Some primitive vessels are present in gnatum and ephedra plants of gymnosperm. So this is the characteristic of the vessels. Now we talk about a function. So when we talk about a function, we get to know that they play a role in conduction of water and mineral, and second, mechanical support. Third is wood fiber or xylem fiber. This wood fiber are nothing but they are the sclerenchymatous fiber. So they are elongated cells with pointed ends on both sides. Thick, extremely thick, lignified cell wall, and due to presence of lignified cell wall, the lumen is obliterated. They have simple pits, so the diameter of pit is same. This wood fiber or Xylem parenchymatous fiber or xylem fiber, they are commonly found in secondary xylem, can be septate or aseptate. So if we see the diagram somewhat look like this. Lumen is almost reduced. And in this you will find a septa, so they can be septate or aseptate. Now when we talk about this parenchymatous fiber, they are of different forms, and the different forms are, first one is fiber tracheids. Fiber tracheids are like tracheids with bordered pit, like tracheids with bordered pits. Whereas second type is of libriform fibers, and these libriform fibers, they are extremely thick walls they have, and they have a simple pits, and they provide a mechanical support. So function of xylem fiber is to provide mechanical support. So that is all about the xylem fiber, and we are talking about the fourth component of this, that is wood or xylem parenchyma. Wood or xylem parenchyma. When we talk about wood or xylem parenchyma, they are a living parenchymatous tissue. They are a living parenchymatous cells, and they are found associated with the xylem. They are living, so they have a thin wall. Protoplasm is present, and that is dense. They have dense protoplasm. Nuclei is present. Okay, they carry out, if we talk about function is storage of food also they carry out, and one more thing, that they carry out radial conduction of water, so known as ray parenchyma. So that is about the wood or xylem parenchyma. So next we will talk about the second permanent tissue, that is a phloem. This phloem is also known as bast or leptome. Now when we talk about a phloem, the function of phloem is to carry out conduction of food. But when we talk about a phloem component, this phloem is made up of, number one, sieve tube, companion cell, third, phloem fiber, and fourth Phloem Parenchyma. Now when we talk about in case of all this, phloem fiber is a dead or non-living and other three are living. So this is living, this is living, and this is also living. Now when we talk about this part, that the sieve tube living, companion cell are living, so we are going to talk about this all four component. In this, sieve tube and companion cell are associated. Sieve tube and sieve tube elements are the cells of this sieve tube, and companion cell are ontogenetically related. So they are known as sister cells. Now if we talk about a sieve tube and companion cell, first we talk about a sieve tube. So sieve tube are made of sieve tube element. They are living, so they are thin walled, okay, but nucleus is absent at maturity, so they are anucleated. So when we draw the structure of this sieve tube, that occurs somewhat like this, and here they have sieve plates. So these are sieve plate, and with this you find this, they are the companion cell. Now in this, protoplasm is peripheral. Why protoplasm is peripheral? Because conduction occurs through this. Now in the sieve plate there are some pores, and through pores the conduction of food occurs from in this way, and companions are associated with this. So now the nucleus is absent in sieve tube element. This one is a sieve tube element, another is sieve tube element, and with that companion cells are associated. So if the nucleus is absent, then the pressure gradient or flow is managed by the companion cell. Okay, so this is the sieve tube element and these are the companion cells. Now, so cytoplasm is peripheral and in thin layer. Now they have P protein known as Phloem protein. They are proteinaceous structure. They are proteinaceous structure present in sieve tube. And are believed to be responsible for movement of material, and number 2, that is sealing of pores after wounding. Now we remember this phloem protein are present at the sieve plate. Apart from phloem protein, we find another deposition, that is callose plug. So at the end of growing season, end of growing season, callose plug made of beta 1,3 glucan is deposited on sieve plate, which inhibit activity of sieve tube. Spring season callose plug get dissolved. So this is what about the particularly sieve tube. So sieve tube, if we talk about function, obvious function is conduction of food and conduction of food specifically. Next, second component of phloem is companion cell, and as we have seen, they are found associated with sieve tube element. They are thin walled, they have a proper cytoplasm. Okay, now when we talk about the function of companion cell, that is important. Companion cell maintain pressure gradient of sieve tube element. It maintains a pressure gradient of sieve tube element. They have a dense cytoplasm and nuclei. Okay, that also you should remember. Now third is phloem fiber or bast fiber. This phloem fiber or bast fiber are the sclerenchymatous fiber. They are nothing but a sclerenchymatous fiber, but they are absent or fewer in primary phloem and found abundantly in secondary phloem. They are a sclerenchymatous unbranched fiber. They are unbranched. Okay, they are dead cell, so protoplasm or protoplast is absent. Phloem fibers of jute, flax and hemp. So the fibers of jute, flax and hemp are nothing but the phloem fiber, and they are extracted for making ropes and coarse textiles. Okay, so that is the dead cell phloem parenchyma or bast fibers. Last component of phloem is phloem parenchyma. This phloem parenchyma are living cells. They are thin walled, they have a dense cytoplasm, cellulosic cell wall, and their main function is storage of food. Now important point that you should remember that they are not found in the monocot plant. So this is the living component. Before that we have to talk about dead component, that is phloem fiber. So now till now in this lecture we have discussed about a simple permanent tissue, that is a parenchyma, collenchyma, sclerenchyma, and the complex permanent tissue, that are xylem and phloem.